Hey guys, this video we're looking at GCSE Biology and we're looking at culturing microorganisms and practical alongside it. So you can grow bacteria in the lab. So bacteria and some other microorganisms are grown, also known as cultured, in a culture medium, which contains the carbohydrates, minerals, proteins, and vitamins they need to grow. The culture medium used can be a nutrient broth solution or solid agar jelly. In the lab at school, cultures of microorganisms are not kept above 25 degrees Celsius because harmful pathogens are more likely to grow above this temperature. In industrial conditions, cultures are incubated at higher temperatures so that they can grow faster. You can grow bacteria in the lab. So bacteria grown on agar plates will form visible colonies on the surface of the jelly, or will spread out to give an even covering of bacteria. To make an agar plate, hot agar jelly is poured into shallow round plastic dishes called Petri dishes. When the jelly is cooled and set, inoculating loops, also known as wire loops, can be used to transfer microorganisms to the culture medium. Alternatively, a sterile dropping pipette and spreader can be used to get an even covering of the bacteria. The microorganisms then multiply. So you can investigate the effect of antibiotics on bacterial growth. You can test the action of antibiotics or even antiseptics on cultures of bacteria. What you do is you place paper discs sold in different types or different concentrations of antibiotics on an agar plate that has an even covering of bacteria. Leave some space between the discs. Then the antibiotics should diffuse, also soak in the agar jelly. Antibiotic resistant bacteria, which is bacteria not affected by antibiotics, will continue to grow on the agar around the paper discs, but non-resistant strains will die. A clear area will be left where the bacteria have died, and this is called an inhibition zone. Make sure you use a control, and this is a paper disc that has not been soaked in an antibiotic. Instead, you soak it in sterile water. Then you can be sure that any difference between the growth of the bacteria around the control disc and around one of the antibiotic discs is due to the effect of the antibiotic alone, and not something weird in the paper, for example. You leave the plate for 48 hours at 25 degrees Celsius, and the more effective the antibiotic is against the bacteria, the larger the inhibition zone will be. You need to use uncontaminated cultures. So contamination by unwanted microorganisms will affect your results and can potentially result in the growth of pathogens. To avoid this, the petri dishes and culture media must be sterilized before use, for example by heating to a high temperature to kill any unwanted microorganisms that may be lurking on them. If an inoculating loop is used to transfer the bacteria to the culture medium, it should be sterilized first by passing it through a hot flame. After transferring the bacteria, the lid of the petri dish should be lightly taped on to stop microorganisms from the air getting in. The petri dish should be stored upside down to stop drops of condensation falling onto the agar surface. You can also calculate the size of the inhibition zones to compare results. So you can compare the effectiveness of different antibiotics or antiseptics on bacteria by looking at the relative sizes of the inhibition zones. The larger the inhibition zone around a disc, the more effective the antibiotic is against the bacteria. You can do this by eye if there are large differences in size, but to get more accurate results, it's a good idea to calculate the area of the inhibition zones using the diameter, which is the full distance across. You don't open the petri dish to measure the inhibition zones, as they should be visible through the bottom of the dish. So to calculate the area of an inhibition zone, you need to use this equation. It's A equals pi r squared, which is also the area of a circle. You're likely to use the unit centimeter squared or millimeter squared. R is the radius of the inhibition zone, and it's equal to half of the diameter. Pi is just a number, so you should have a button for it on your calculator. If you don't, you just use the value 3.14. Here's an example question. So it says the diagram below shows the inhibition zones produced by antibiotics A and B. Use the areas of the inhibition zones to compare the effectiveness of the antibiotics. The first part is to divide the diameter of zone A by 2 to find the radius. So the radius is 14 divided by 2 to give us 7 millimeters. Then you stick the radius value into the equation area, pi r squared. So area of A is pi times 7 squared, gives us 154 millimeters squared. You then do the same thing for zone B, and you get the area of zone B equals 314 millimeters squared. Then you compare the sizes of the areas. 340 millimeters squared is just over double 154 millimeters squared. So you can just say that the inhibition zone of antibiotic B is roughly twice the size of the inhibition zone of antibiotic A. The equation can also be used to calculate the area of a bacterial colony. You just need to measure the diameter of the colony you're interested in first. So here's some quick recall questions to try. What is the maximum temperature that microorganisms should be grown at in a school lab? Why shouldn't a temperature above this be used? Then there are ways in which you can make sure an experiment testing the effect of antibiotics on bacteria has not been contaminated. Give 3 of these ways.